When you think about liquid biopsies, those amazing blood tests for cancer, your mind probably goes straight to DNA, right? Circulating tumor DNA, ctDNA, we hear about it all the time.
Those bits of genetic code floating around.
Exactly. But today, we're diving deep into something else, the sort of original star, circulating tumor cells, CTCs.
That's right. CTCs are, well, they're whole cancer cells, actual cells that have broken off a tumor and gotten into the bloodstream.
Okay, so whole cells, they were actually the first idea for a liquid biopsy.
There were, yeah. But for years, honestly, they got overshadowed, mostly by ctDNA.
Why? I mean, why would the whole cell lose out to just fragments? Seems a bit backwards.
It really boiled down to practicality. ctDNA, once you spin down the blood, it's relatively easy to isolate from the plasma and sequence.
Okay.
CTCs, though, being intact cells, finding them, purifying them, much, much harder. So our mission today is to figure out why CTCs are making such a strong comeback.
So new tech.
Exactly. New bioengineering is letting us finally capture these things in decent numbers. And they're revealing unique stuff, especially about drug resistance, how cancers metastasize things ctDNA alone just can't show us.
Okay, let's get into that. The core problem seems to be just how rare they are. How long have we actually known about these cells, and why are they so hard to find?
It's kind of amazing we've known about them for a very long time, nearly 2 centuries.
Wow, really?
Yeah, a surgeon, Thomas Ramsden Ashworth, first described them way back in 1869, saw them in a patient with metastatic cancer. But the rarity, it's extreme.
How extreme are we talking?
Well, think about this. A tumor might shed millions of these cells every single day for every gram of tissue. Millions.
Okay, that sounds like a lot, so they should be easy to find.
You'd think so, but they get diluted in, liters of blood. The key statistic, the real challenge, is you might be looking for just one single tumor cell mixed in with maybe 10 billion normal blood cells.
1 in 10 billion. That's, I can't even picture that. That's like finding one specific person in the entire world population almost.
It's staggering, isn't it? And even with current standard methods, like the FDA-cleared cell search system.
That's the main one people use.
It's the benchmark, yeah. It typically finds maybe 1 to 10 CTCs. That's in a standard blood draw, like 7.5 milliliters. And even then, only in about half to maybe 80% of patients with advanced cancer.
Finding, 5 or 10 cells. Okay, maybe that's useful for just counting them, saying yes, they're there, or no, they're not. But for doing deep analysis, sequencing, omics, that sounds totally insufficient.
It barely scratches the surface for that kind of work.
Yeah.
Precisely. And on top of that, the way we used to identify even those few cells, it was biased.
How so? What was the old definition?
The classic definition was based on staining. The cell had to have a nucleus, be negative for a marker called CD45 that rules out white blood cells, but positive for epithelial markers called cytokeratins.
Okay, so basically, looking for cells that looked like they came from the lining of organs, the epithelium, like they're wearing an epithelial uniform.
Exactly. But here's the catch. When cancer cells get really aggressive, when they want to move to metastasize, they often undergo this change called epithelial to mesenchymal plasticity, EMP or EMT.
Right, they sort of shed their old identity to become more mobile.
They do, and part of that shedding involves losing those very epithelial markers like cytokerations. They ditch the uniform.
So the classic definition misses them.
It misses them, and that's a huge limitation. It doesn't just miss CTCs from cancers that aren't epithelial to begin with, like sarcomas or melanomas. Critically, it misses the potentially most dangerous cells, the ones that have made that EMT switch and are geared up for invasion.
Okay, so if that classic definition is biased against the really important metastatic cells, how do researchers now say, okay, this cell is definitely cancerous and not just some weird-looking normal cell? Are those old methods basically useless now for finding the aggressive ones?
They're definitely limited, biased, which is why the field has shifted. The sort of bona fide definition now relies on something more fundamental, genetics. Specifically, chromosomal aneuploidy, or CNV.
CNV, copy number variation.
Exactly. These are big changes in the chromosomes, large chunks gained or lost. It's a hallmark of cancer, a real signature of malignancy that normal cells just don't have.
So instead of looking at the cell's surface uniform, you look at its messed up genetic blueprint. That tells you it's gone rogue, regardless of what it looks like on the outside.
Precisely. It gets around all the issues with staining, whether the cell is epithelial or mesenchymal, all that bias. If it has significant aneuploidy, it's almost certainly a tumor cell. Identifying CNV at the single-cell level is now the gold standard for confirming you've actually got a CTC.
Right, that makes sense.
Yeah.
But that brings us back to the tech problem. If you need to do that kind of detailed genetic analysis, you don't just need 1 or 2 cells confirmed. You need lots of them, and they need to be pure. What level of purification are we talking about, starting from that 1 in 10 billion soup?
The enrichment needed is, well, it's astronomical. And it depends what you want to do downstream. For some simpler bulk analyses, maybe like digital PCR, you might get away with, say, 1,000-fold to 10,000-fold enrichment.
Okay, 103 to 140 soup, still a lot.
It is. But for the really detailed single-cell stuff, the omics, looking at RNA, proteins, the full genetic landscape, you need purity that's greater than 100 million-fold, 108-fold enrichment.
Hold on, 100 million-fold. Why does single-cell analysis demand such an incredibly high level of purity? That sounds almost impossible.
Think about it. You're trying to analyze the tiny amount of material inside one single cell. If you have even a few contaminating normal blood cells, especially white blood cells, which are packed with RNA and DNA, their signal will completely overwhelm the signal from that one rare CTC.
Contamination swamps the signal.
Completely. You're looking for subtle changes, specific mutations, gene expression patterns unique to the cancer cell. Any significant contamination makes your data essentially meaningless. Garbage in, garbage out.
Okay, so how on earth are scientists achieving 108-fold purity? We know the standard positive selection, grabbing cells with EpCAM, is biased. What are the newer tricks?
The strategies generally exploit differences, molecular, immunological, physical properties. Since positive selection, like EpCAM, has its issues.
Like missing the EMT cells.
Right. The less biased approach is negative enrichment. Instead of trying to grab the rare CTCs, you get rid of everything else. You deplete the billions of normal blood cells, usually using antibodies against common markers like CD45.
So you throw away the normal stuff and keep what's left.
That's the idea. But doing that efficiently enough to get that 108, 101N purity, it often requires really clever combinations of methods or highly advanced microfluidic devices that can process cells incredibly fast and selectively.
But still, even with clever depletion, you're limited by the starting volume, aren't you? That 7.5 millileb tube.
Yeah.
It just doesn't have many cells to begin with, even if you could purify them perfectly. Where did the real breakthrough come from to get the numbers needed?
That's the crucial point. The key technological leap was finding a way to process much, much larger volumes of blood, breaking that biological bottleneck of rarity. And that's done using a clinical procedure called leukapheresis.
Leukapheresis. Isn't that used for, like, collecting white blood cells for donation?
It is, yes. Or sometimes removing excess white cells in certain diseases. But here the principle is adapted. Instead of a small tube, the patient's blood is circulated through a machine that processes maybe 1 to 2 liters of blood over a few hours.
1 to 2 liters, compared to 7.5 milliliters. That's a huge difference.
Massive. So you take that leukapheresis product, which is enriched for white cells, but also contains any CTCs that were in those liters of blood, and then you run that through a high-throughput, super-efficient depletion system, often microfluidics-based.
Okay, so big volume in, run it through the fancy filter.
Yeah.
What's the yield like then, compared to the 1 to 10 cells we talked about earlier?
It's transformative. Using this approach, researchers have been able to isolate anywhere from 100 up to 58,000 CTCs from a single patient sample.
58,000. From one patient.
From one leukapheresis draw, yes.
Yeah.
It completely changes the game. Suddenly, you have enough cells not just to count, but to do deep single-cell sequencing, proteomics, functional studies. You can finally start asking really detailed questions about what these cells are actually doing.
That's the perfect lead-in. Now that we can actually get our hands on thousands of these cells in high purity, what have they taught us? What secrets have they revealed about metastasis, about how cancer actually spreads through the blood?
Well, we've confirmed CTCs are the key players in spreading cancer via the bloodstream hematogenous dissemination. The whole process involves steps like invading out of the main tumor, getting into a blood vessel that's intravasation.
Freezing in.
Right. Then surviving the trip through the circulation, which is surprisingly tough for them. And finally, getting out of the blood vessel in a distant organ extravasation and trying to set up shop as a new tumor, or what we call a disseminated tumor cell, a DTC.
You said surviving circulation is tough. What's the survival rate like for these cells once they're in the bloodstream? It sounds like a dangerous journey.
It's incredibly dangerous for them. The survival rate is shockingly low. Remember those millions shed daily? Fewer than 1%, probably much less, maybe 0.01%. actually succeed in forming a metastasis.
Wow.
Their average half-life in the blood is unbelievably short. We're talking seconds to minutes.
Seconds, not hours or days.
Seconds to minutes. The bloodstream isn't a gentle river for them. It's more like a high-speed, hostile environment. They face physical damage from sheer stress. They risk dying because they're detached from their normal environment, a process called anoikis.
Like homelessness for cells.
Kind of, yeah. And of course, they face constant attack from the immune system. Most CTCs are eliminated almost immediately.
Seconds to minutes. They really are just fleeting passengers fighting for dear life.
They are. And that fight, that vulnerability, probably explains why the next finding is so important. CTC clusters?
Clusters, you mean they travel together?
Exactly. While most CTCs seem to travel alone, a fraction, maybe 1%, up to 30% in some studies travel as clumps, as multicellular clusters. And these clusters are bad news. They show a dramatically higher potential to form metastases.
How much higher?
Something like 25 to 50 times more metastatic potential compared to single CTCs traveling alone.
25 to 50 times. That's huge.
Yeah.
Why would traveling in a group be so much more effective? Is it just safety in numbers?
That's a big part of it. Being in a cluster seems to offer physical protection, makes them less susceptible to anoikis. Maybe cells on the inside are buffered. It might also help them evade immune detection.
Makes sense.
And interestingly, these clusters aren't always just tumor cells. They can be heterotypic. Meaning they can actually recruit and travel with normal cells like platelets, certain immune cells like neutrophils, or even fibroblasts. These hitchhikers might actively help the cluster survive the journey and maybe even help them settle into the new organ.
Wow, like a little invasion squad.
Oh.
Yeah, that's fascinating. Now let's shift to heterogeneity. You mentioned single-cell analysis is key here. What's the aha moment when you sequence thousands of individual CTCs from one patient? What do you learn that you'd miss from just looking at one biopsy from the main tumor?
You learn that the cancer isn't one single entity. It's incredibly diverse, dynamic. Single-cell RNA sequencing a cRNA-seq has just blown the doors open on this. It reveals massive differences in gene expression from cell to cell.
Could you give an example?
Sure. In prostate cancer, for instance, when patients become resistant to standard anti-androgen therapies, researchers used cRNA-seq on CTCs. They found specific subclones of cells had activated a pathway called non-canonical WNT signaling, and that seemed to be driving the resistance, something you wouldn't necessarily see clearly in a bulk analysis. We also used those CNV markers, the genetic signature,
The aneuploidy.
to identify really weird cells that the old definitions missed entirely. We found these small cells that didn't have CD45 or cytokeratins, so-called double negatives, but their CND profile proved they were malignant. Turned out they were neuroendocrine-like cells, which are known to be very aggressive in prostate cancer.
So you found a hidden aggressive cell type just by looking at the genetics.
Exactly. And in breast cancer, scRNA-seq has captured cells literally switching states, showing dynamic plasticity. Some cells might be focused on proliferating, driven by HER2 signaling, while others switch to a more dormant, resistant state driven by Notch signaling, all within the same patient at the same time.
That implies the tumor isn't static at all. It's actively changing its strategy, maybe cell by cell. So analyzing CTCs gives you a kind of real-time snapshot of the cancer's current game plan, revealing these functional subclones that might be hiding out, missed by a single biopsy taken months ago.
That's precisely the value. It's a real-time window into the tumor's evolution. We can spot resistance mechanisms as they emerge. For example, detecting the Edd EFR T790M mutation in CTCs from lung cancer patients tells you they'll likely stop responding to certain targeted drugs, or finding the ARV7 variant in prostate CTCs.
What does ARV7 signify?
It's a variant of the androgen receptor, and its presence strongly predicts resistance to common prostate cancer drugs like enzalutamide and abiraterone. Finding it in CTCs is a clear signal that the treatment needs to change.
Okay, let's connect this to the clinic. With all this information, what are the main ways CTCs are being used right now or poised to be used? And how do they fit alongside ctDNA going forward?
Right now, there are two main clinical applications emerging. The first is prognostication and enumeration, basically counting them.
Just the number matters.
The number definitely matters. Consistently, higher CTC counts correlate with worse outcomes. In metastatic breast cancer, the established cutoff is 5 or more CTCs per 7.5 milliliters of blood. Patients above that threshold generally have poorer survival, and watching the count over time, if it stays high or keeps rising during treatment, That's a strong indicator the therapy isn't working.
Okay, so counting for prognosis, what's the second use?
Molecular monitoring. And this is where CTCs offer something unique compared to ctDNA.
How so?
ctDNA is great for looking at the DNA blueprint, finding those known mutations we talked about, but many new therapies target proteins, especially proteins on the cell surface.
Like antibodies or CAR T cells.
Exactly. Things like antibody-drug conjugates, ADCs, which are like guided missiles carrying chemo, bispecific T-cell engagers, BiTEs that link immune cells to cancer cells, and CRT cells. For these therapies to work, the target protein actually needs to be present on the surface of the cancer cells.
And CTC analysis can tell you that.
Yes, because you have the whole cell, you can do epitope assessment. You can actually look and see if the target protein is there on the surface, ready to be engaged by the drug. ctDNA can't tell you if a protein is being expressed and displayed, only if the gene for it is there.
That's a crucial difference for protein targeting drugs.
It really is. Plus, since CTCs are shed from potentially all tumor sites, the primary, the metastases analyzing them gives you a kind of summative representation of the entire disease burden. It addresses that problem of tumor heterogeneity better than taking a biopsy from just one metastasis. We mentioned ARV7 in prostate cancer CTCs predicting resistance. That's a prime example of molecular monitoring guiding therapy right now.
Got it. And what about the future? Early detection or detecting minimal residual disease MRD after treatment?
That's a huge area of interest. We already know that finding CTCs, even in early stage, seemingly localized cancers, like in about 20% of stage I to third breast cancer patients, is linked to a higher risk of the cancer coming back later.
So their presence signals hidden danger.
It seems so. The future for MRD is likely combining the strengths of both ctDNA and CTCs. Use ctDNA for its high sensitivity to detect tiny amounts of tumor DNA, confirming our D is present. Then, analyze the CTCs found, even if very rare at this stage, to understand their biology. Are there specific resistance mutations? Are they expressing targets for therapy?
So ctDNA says something's there, and CTCs say, here's what it is and maybe how to attack it.
That's the potential. If you can identify actionable targets when the tumor burden is at its absolute lowest, maybe you can guide interventions, perhaps adjuvant therapy, that could actually be curative, preventing recurrence before it really takes hold.
It really feels like CTC analysis, powered by these amazing bioengineering advances like leukapheresis and single-cell tech, is moving way beyond just being a biological curiosity.
Absolutely.
ctDNA might be simpler for getting that basic genetic blueprint, but it seems CTCs offer this richer, more functional picture. The cell state, its protein landscape, the clonal diversity. That seems vital for guiding the really sophisticated targeted and immune therapies we have now and the ones coming down the pipeline.
They really are complementary. You get the genetic code and the functional reality. Having both pieces of information is incredibly powerful for making the best treatment decisions. That's really the key takeaway.
Okay, so to wrap up, let's leave our listeners with something to think about. We learned that the average lifespan of a CTC in the bloodstream is incredibly short, just seconds to minutes. That means the blood is this highly vulnerable transit point for cancer cells. So here's the question. What if we could focus treatment specifically on that fleeting window? If we could therapeutically target CTCs while they're circulating, maybe exploit unique weaknesses they have during transit, like specific metabolic needs or survival pathways, could we effectively stop metastasis before it even gets a chance to seed? Could we shift cancer treatment from reaction towards actual prevention of spread? Something to ponder.